Hi, welcome,
Hi,
Frederic. Nice to see you.
nice to see you again.
Yeah, thank you so much for joining. I really appreciate it. Let me do speaker. Um, we have had such wonderful conversations as part of these career conversations, and yours is such an interesting journey into the work that you do. Can you introduce yourself and where you're working now and what you're busy doing?
Yes, absolutely. My name is Frederic Vigneault. I'm a senior staff scientist at the Wyss Institute, which is a translational research institute at Harvard University. I've been there for 10 years. I work on many different projects. My background is in molecular biology. And at the Wyss, I started first with George Church, like working on genome engineering in bacteria, then next-gen sequencing, later move on to CRISPR, so gene editing in the mammalian cells and human cells. And now I'm also doing that in tadpoles and like frogs, basically. I also work on diagnostic companies with Jim Collins, like Sherlock Bioscience. We made the news during COVID, and that's my journey at the Vyss in a short amount of time,
In a
10 years.
short,
Yeah.
yeah, that's all the big terms, all the big folks, all the heavy hitters in the science, engineering, and in this field. So pretty amazing stuff. I guess, do you have a favorite of all the things that you're working on? Or I think tadpoles is incredible that you're doing this in tadpoles, but what do you find the most exciting or interesting?
Well, tadpole is the one I'm currently doing now. I think it's very interesting because we're using it as a model to for cognitive disorder. So we're trying to model rare diseases. The one we've picked on as a first try is Rett Syndrome, which is a rare disease of neurodevelopmental disorder. So there's like several symptoms from learning disabilities, seizures, breathing. It's pretty severe. So we started using Tadpole as a way to model it quickly. So instead of having to spend months or maybe like even a year developing a mice model, we can do it in just a couple of weeks. And in a couple of weeks, we can also start screening for compounds that can address those symptoms. And it turned out that the tadpoles, because they have a lot of similarities in our genes, the brain structure, they also have lungs. There's a lot of things we can screen for. We can also train them. We can also look at sleep disorders. So yeah, so we can basically in a couple of weeks can just start screening 1,000 tadpoles for like 10, 20, 30 different drugs, figure out which one is the best. It's something you cannot do with cell culture. With cell culture, you cannot train for a cognitive disorder like behavior. And something that's very hard to do with mice just because of the scale. It's hard to scale mice, like 1,000 mice, nobody does that. Typically, you're going to have one drug. You're going to try it on like 20, 30 mice, and then, it's hit or miss. You try again.
Right. You seem to have found that Goldilocks space, right? Cell culture, all very interesting, but it's very limited in terms of what you can find. Mice, very cool, but you can only do so many and they're slow. So tadpoles are kind of in the middle. There are lots of them and they go pretty fast if you can get the breadth of the application space in them. That's very, very interesting stuff. Cool. And a lot of applications, a lot of health applications for this. Are you thinking forward in terms of what this would, look like if you're wildly successful?
Yeah, so we've been coupling this with a prediction, like a computational prediction. So we bring the machine learning to predict what drug's going to work with this like biological model screening. And so we did that for Rett syndrome, and were sort of like the current best candidates. We tried it in the mice model for Rett syndrome, and it worked well. So now we're actually looking to bring it to a clinical trial in the next year or so. And if that works from there, we are thinking to start doing other rare diseases. So we're thinking to start a company to do that. Yeah.
Amazing. So not only like all the heavy hitters in terms of molecular biology, genetic engineering, synthetic biology, and all the amazing labs doing it, but now also bringing in AI and clinical trials and entrepreneurship. So you run the gauntlet. You've got the whole the whole package going. So that's very cool.
I mean, there's a lot of that at the Vyss. It's pretty resourceful institution, a lot of funding, a lot of big PIs. So a lot of different disciplines too. So putting different people together like to try to give like a different approach to like a current need. Something that's different that let's say the pharma company might have had a trouble getting into like in general, when you look at like Alzheimer, Parkinson, they're very tough disease to go after. So maybe at one point that's where we're gonna aim. But yeah, so we're trying to have like a different angle. Hopefully that angle is gonna be helpful.
Very cool. Yeah, no doubt. I think when you put people with different perspectives together, there's ways of thinking that any individual might not have had the ability to do. Not a lot of people may know about the Wyss. Can you just say a few words about where it is and what it is, and a little bit?
Yeah. So the Wyss Institute is part of Harvard University. We're located in the Longwood Medical area, so Harvard Medical School. It was started in 2009. So I joined in 2010, like a year after it started. And the goal of the Wyss Institute is translational, which means you take academic research and you try to de-risk it so it can become like a product or a company or being licensed to another company. That's basically the goal. And I've done that a few times. I have several patents, and they've all went to different companies. Yeah.
Wow. So you are a scientist, engineer, entrepreneur, forward-looking thinker. Did you start off this way? You didn't. I mean, the Wyss is a new institution. How does one start if you're looking at landing at a place like the Wyss?
It's interesting. So in my case was through networking. So I did my PhD. So I'm from Quebec, in Canada, and I did my PhD there. I actually did it in plant biotechnology. And instead of doing a postdoc in academia, I tried to look for a postdoc in industry. And it took me a while. You just apply, you go to conferences, you present, you meet people. And after 6 months like that, I found a place I call SRI International, which is it's a nonprofit research institute, but they're really big on research institute. People using Siri that comes from SRI basically. They're a powerhouse in terms of innovation. So that's where I came from originally. With doing mostly computational. So even though my background is like really much wet bench biology, they wanted biologists to help them with the data curation, which what we call data analyst these days. But like 12 years ago, data analyst wasn't as trendy as it is today. So data curation, they were using AI, wasn't as trendy, was just getting started. Yeah. The Wyss Institute started. I learned about it from different people. They like told me about it, so I applied, got an interview, went well, and
and the rest has happened, as you say. Right?
Yeah, networking is basically the best way to do about that. And if you're looking for people actively, like going to conferences, meetings, and like talking to people, then that's how you make your networking happen.
Yeah, I get that question sometimes, like what courses should I take or what should I be doing if I want to become a synthetic biologist or an entrepreneur or an AI, data analyst or whatever you'd like to call them now, right? So, is there a recipe? Networking is the way you sort of bring yourself forward out into the community, but is there a background that you think is particularly helpful for getting going?
I don't know. When I got my interview at SRI International, my background was in plant biotech. They didn't do any plant biotech at all. They were looking for a position mostly on infectious disease. So the way I did it was to really tailor my presentation. So the same I've been giving to other people, but instead of that, I tailor. So you have like your PowerPoint slide, and on the side I put like a plant model or adaptive that we're using, because I didn't expect them to know all the parts of the plant. And I was doing molecular biology, like on the kinases that helps with plant growth. So basically every time I'd be somewhere, I'd be pointing toward the plant, exactly what it is. I give them some context. Mm-hmm. And I finished something I was interested to do, but I didn't really do as part of my PhD, is like a network analysis, like a signaling cascade, if you're like this variant binds to this one or affect this one this way. It's something I like to do. And because I was working with kinases, they kind of do that. They're signaling molecule in your cell. And the job application was for doing this data curation for making network. So I basically created a network from, like, I just draw it, like from literature, from what I know, from my own finding, like I found this, so this means that this network is going this way and not this way. And they loved it, right? I tailored it to what they wanted to know.
Yeah.
And they said I was the only one who did that, right?
Yeah.
So, you know,
it's such great advice, because I think what it shows is that you put yourself into the mind of the person who is watching you, right? A lot of times people think that they have to present themselves and it's about them, but it can be very successful to think in the reverse. Like, if you were sitting in the audience, what do you need? What do you want to know? What would be helpful? And when the presentations meet all of those things, there's an instant connection and a, like, you know, you feel like it could be a win by, you know, working more with that person. I think that's so wise.
and it's not always easy, right? But when I got my interview at the V's, they were looking for people with a private sector, like 2 years of experience in private sector. So, you know, had that. That was basically the main thing they were looking at. Plus, like, if you're experienced or interested with the current project. But trying to put yourself in the mind of George Church is not possible. So, let me tell you. So,
It's a scary place. We don't want to go there.
No, and my feeling of what is possible was already pretty high, so I think that's why it went well. George doesn't have a ceiling to a point where sometimes I think like it's like, yeah, you're taking it this way, but that's not possible. That's like, we'd have to invent new physics type of things. And you've done that before. You know, those things fail all the time. You know, they're
Right,
to me,
right.
they're predictable. But the ceiling, your ceiling has to be really high. Whether you honestly want to meet with George Church as a scientist or like a future scientist, things that we can control or would like to control one day, they're pretty high. So my feeling is basically, if it's possible in this universe, then you know, it's possible. That's it. So if you can like sort of make the step to get to that point.
Yeah.
I remember when I started biology, people were sort of mocking the idea of to do like a brain transplant, and it's been tried more or less successfully. But you know, they tried it on a marmoset, and like to transplant a head, and the marmoset survived for a few hours, right? So, and was cognizant the whole time. So not a total success, but you know, not a total failure either. And but you know, when I started biology, people were saying it's impossible. But I was a believer. I actually believe that. Now I believe it's completely pointless to do those type of things. But you know, that's different. It's a different story.
Yeah, I think that the goalposts keep moving, and I think the things that we would have thought were impossible, we take as a given right now, right? We just sort of accept as truthful and part of our world. I certainly think a lot about that with computers, right? So what people imagined computers would be used for and we're capable of is very different than what we are living with right now and where computers are in our lives. So biology as the next frontier, I'm hopeful, is going to be in a similar boat.
Yeah, I mean, you think about longevity, by example. You know, now we're not saying like the thought leader, like including George, in the field of longevity, they're saying like aging, they're sort of treating aging as a disease. So you know, it's a biological process where all our biological processes are becoming older here and there. So maybe we can fix that. And because it's in the realm of physics, you know, in theory, it's possible that we may be able to extend someone's life or make someone younger. We're still really, really early, but you know, it's a hot field. And on the computer side, there's actually, people have devised AI a long time ago, like back in the 60s. It's just we didn't have the computational power. And the same thing's happening with biology, like just in CRISPR now. It makes making models like tadpole models so much easier. Like we can make, like it takes me a day to design something, order it from a company. It takes them longer to ship it than for me to do everything else around it, right? So yeah.
Isn't that incredible? The ability to accelerate biology and biological research and engineering is just, it is the world we are looking towards. So that's a great thing. We need that for sure. So
yeah.
Amazing. So so if there was a very excited 17-year-old student watching this, what would you advise that student?
I mean, my experience with BioBuilder has been like with having an intern. I just had this like I had him like 2 years in a row. So like really like get out there, try to find like a place where you can just do science. I mean, you're you're building yourself by your experience, right? So if you want to become a scientist, then do science. That's
Absolutely. Here, here. I couldn't agree more. Yep.
yeah.
Yeah.
And you know, so with Brian Zetto, he started, he knew a little bit about PCR because, you know, you've been telling them about that. And my whole internship was like, yeah, you're going to need to do PCR. So basically he spent a bunch of summer learning about PCR, designing, and eventually even went to real-time PCR. And he came back with me the year after. He was now like a Boston University student. And so yeah, we're gonna like you're still gonna do PCR, but this time I need you like to assemble pieces. So now I started doing like Gibson assembly, all those kinetic construct that we can make. It was great. Yeah, I had a really good time with Brian. And super shy person, especially the first year. The second year was much better. And so I don't know exactly what he's doing. I mean, it's COVID. They probably stayed home. Hopefully, stayed home.
Yeah. Yeah. We'll follow up. We'll make sure we do a little reunion because it has been
But
wonderful having you
yeah,
in the BioBuilder community, mentoring the students and teaching them what it is like, as you say, you know, if you want to be a scientist, do science and learn as you go. And I think that's a great bit of advice.
and he did mention it was helpful to him during his first year because there was a lot of things they were learning about, and he said, "Yeah, I've done that." So
Right, isn't it? It does. It positions you well. I'm very glad to hear that. Well, thank you so much for sharing your story. It's fascinating. I cannot wait to see what all this leads to with you. I'm just, it's going to be so exciting.
yeah.